Into going into chemistry. So let's kind of review. Hopefully we'll get through it quickly. We have a lot of slides, but they aren't really too busy with a lot of information. So, all right. So the structure of atoms. So chemistry, we're going to be talking about the interactions of atoms with each other and with molecules. The atoms will unite together to form molecules. So the atom is the smallest unit of matter and it can't be subdivided. It does have its subatomic particles, but those can't be divided really, to make it a smaller substance that you're changing what your substance is when you have different amounts of protons and neutrons. All right, so the atoms will interact to form molecules, okay? They'll form bonds to each other, and we'll talk about the different types of bonds that are formed to make the molecules. Atoms are composed of the electrons, the protons, and the neutrons. So your protons and your neutrons are in the nucleus or the core of the atom, okay? And your protons are positively charged, all right? And your neutrons are not charged, neither positive nor negative, neutral, not charged. Your electrons are the negatively charged particles. And these are going to be the particles that kind of whir all around about the core of the atom, all right? So here's our picture or structure of the atom, okay? With your protons positively charged, your neutrons have a big circle in it as no charge, and your electron as negatively charged. And you have to remember that these atoms are going to move, these electrons are going to move in shells, and they're kind of going to move and whir all about, they're constantly in motion around the core of the nucleus of the atom, all right? Remember that we have to balance our charges, and that like charges will repel each other and that opposite charges will attract each other, okay? And that'll be important for forming our bonds. All right, so when we fill up our atoms, we fill up with protons and neutrons in the core, and we'll fill up our electrons in valences or shells. And we fill up our interior valences first. So when we get our first two electrons, they go in what's called the S shell, which takes two electrons only, all right? And then the subsequent or outer shells that are higher energy going out here, these will take take 8 electrons in the outer shell. I'm looking at 4 sitting here. All right, so these will be negatively charged moving around. Atoms that have the same number of protons are the same chemical element. So the number of protons is the only thing that determines what the chemical element is, all right? You can add in or take out neutrons, but if the protons stay the same, you got 1 proton in there, you got hydrogen, okay? So you got 6 protons in there. We'll figure out what that one is too. I know what it is, but all right. Each different chemical element will have a different number of protons. And as you go across the periodic chart, you'll add in 1 proton. So moving across the chart, and we have a periodic chart coming up. So your first chemical element is hydrogen. Hydrogen has 1 proton in it. You move across the periodic chart over to helium. Helium will have 2 in it. You drop down to the next row. The third one is lithium. It has 3 protons in it, okay? So as soon as you change the number of protons, you change the atomic number, you're also changing what element you're talking about. That's atomic number. Your atomic mass is how much each atom weighs. And the weight of the atom is made up by the protons plus the neutrons. Okay, so the neutrons doesn't change what element it is, it just changes how much it weighs. And you can have a variable amount of neutrons in the nucleus. Got to have that same number of protons to stay with the same element. So I have here If we have oxygen 15 has 8 protons. Having 8 protons is its atomic number. It's what makes it oxygen. So we know the other thing contributing to its weight is the neutrons. 8 plus 7 is 15, or conversely, you could say 15 minus the 8 protons would tell me that with oxygen 15, I have 7 neutrons. Oxygen 16, which is the common oxygen that we think of, has 8 protons and 8 neutrons in it. So what do you think the composition of oxygen 17 would be?
8 protons and 9 neutrons.
And 8 and 9 neutrons, right. And electrons, where did I leave out electrons in all this? Do you know why I left out electrons?
They have the same amount as the protons.
Well, they typically should balance in their charges the amount of the protons, but electrons are so lightweight, their weight is negligible in terms of the weight of the protons and the neutrons. So we don't even add it into it, because if like a proton were to add, 1 atomic mass unit, an electron is like, a 10,000th of that. Okay. All right. So we don't add it in. All right. So when we're talking about these isotopes, which is what we're talking about here when we talk about the different oxygens, 15, 16, and 17, 16 being the common one, we can talk about whether it's a heavy isotope, like oxygen 17 had additional neutrons in it, 9 neutrons, okay? So a heavy isotope is just simply, an element or an atom that has an extra neutron, maybe it has 2 extra neutrons in it, while maintaining a stable nucleus. It's not going to try to lose the extra neutrons. It's stable living that way. It's just simply added weight to it, okay? But radioisotopes are the other type of isotopes. And these are when your isotope has extra neutrons in it, and that renders the nucleus unstable, okay? And so when you have unstable, as in when we get into carbon 14 here. So typical carbon is carbon 12, has 6 protons, has 6 neutrons in it, okay? Heavy carbon, carbon 13, just has an extra neutron in it. And that's just heavy, it's stable, okay? It has no real need to eject that extra neutron. But when you get up to carbon 14, this is what we use to date things like the dinosaurs, okay? And carbon 14 is going to start trying to get rid of that extra unstable neutron that makes the whole nucleus unstable. And that's what we call leading to radioactive decay, okay? So it's trying to eject that and get it out of there. And we use these radioisotopes, like I said, to do the dating of things like the dinosaurs and that, but we also use it pretty often in medical, in radiography, okay? So any of you who want to go into radiology, you'll go in there and you will oftentimes administer some sort of radioisotope to people and then take pictures. And instead of like getting the X-rays coming from outside of the person and zapping them, the person has the radioisotope traveling around through their blood, and it emits the radiation from inside, and you can take pictures like a PET scan. This is the way a PET scan works, okay? We're not going to get too in depth with this. We do this a little bit more when I teach anatomy, so that you can understand the images. We don't really do the images so much in here, but we sometimes use radioisotopes in microbiology to see where we might put some radioisotope on a microbe to see where it's being taken up, and then we might want to track it in the body to see where the radioisotope is taken up. So let's say I put something that had a radioactive nitrogen in it, okay? Let's say I had a radioactive nitrogen and I put it in my microbes, and then I had it I gave it to a person or a lab rat or something. And where do you think in the body radioactive nitrogen might show up?
Is it in the urinary system?
Urinary system would be a big place it would probably show up because you have all that urea, and that's where you get rid of all your nitrogenous waste. So that's a good answer. You know, and it would probably show up, it would probably get incorporated into some proteins. Okay, so it might also get incorporated into, say, like a lot of your muscle cells, because your muscle cells are primarily proteins. So things like that. So you can think that way when there's questions about radioactive tracers. That's how you think about it. If you put radioactive sugar, you know, into the brain, you know, you could track it for organs that use a lot of sugar. And sugar, as you know, is the primary food that your brain uses, glucose. So oftentimes we use radioactive glucose in PET scans when we want to scan the brain and see what areas of the brain are lighting up to look for things like a brain tumor. Okay, so that's how those questions typically work. All right, so we have the different hydrogens, oxygens, and carbons. So hydrogen has 1 proton, so H1, how many neutrons does it have?
1.
Well, if 1 proton, and I should have said this. So 1 proton always weighs 1 atomic mass unit. 1 neutron always weighs 1 atomic mass unit, and 1 electron is always negligible. So if hydrogen 1 weighs 1, it has how many neutrons?
0.
0, because it has 1 proton. Hydrogen 2.
1 neutron.
1 neutron and 1 proton. Hydrogen 3.
2 neutrons. 1 proton.
1 proton, 2 neutrons. Oxygen 16 has 8 protons, oxygen. That's its atomic number.
So 8
We'll have 8
neutrons.
neutrons.
8 neutrons. Oxygen 17.
9 neutrons.
And oxygen 18.
10 neutrons.
Yeah, there you go. So you have the idea. All right. These are all the elements that you need to be concerned about in biology. So hydrogen, these are pretty much also the same elements that you're going to get concerned about if ever you take organic chemistry or biochemistry. So hydrogen, carbon, and nitrogen. These are the big 3, okay? Big 4, oxygen. And then we have what ends up being a little bit more minor elements, but they aren't that minor because everything, as you'll see in our bodies and in nature, is solvated in water, but it's never just solvated in like kind of pure water. We're salty water. Remember, in evolution, we crawled out of the sea, got some legs, got some lungs that were working, right? And so we're basically sacks of seawater. So we have lots of sodium as kind of our primary element that's floating around in our water, but also with some magnesium, phosphorus, and potassium. So there's sulfur, chlorine, potassium, calcium, iron, iodine on the second slide here. So these are sort of the minor elements. These are the inorganic elements that will form the salts that are in our body. All right, so let's get back to our electronic configurations. So now let's talk about electrons. So our electrons are arranged in shells according to different energy levels. We always fill up the first shell first, and that carries 2 electrons, and that innermost shell is called the S shell. You don't really need to know that, okay? Then the next shell outside of that will take 8 electrons, and we fill that up. Okay, and then we go to the third shell beyond that, takes another 8 electrons, okay? The absence of having some electrons in the outer shell makes that shell reactive. That shell is going to want to be filled up to make it stable, to make it look like what we call the noble gases. The noble gases are all filled in their outer shells. And when that outer shell is empty or has a position empty, it's looking to fill it. It's looking for a partner. And so it's chemically reactive or chemically unstable until it fills that outer shell. Now, also with our electronic configurations, we built up our hydrogen, right? Hydrogen has 1 proton in it, typically no neutrons. Our hydrogen has that 1 proton, so it has 1 positive charge on the inner core of its atomic nucleus. So how many electrons are usually with that hydrogen?
1.
1 electron. And it balances it exactly so that you have the 1 negative charge of the electron to balance the positive charge of the proton. So that's where you think of how the electrons fit in. And usually you'll have an exact match of electrons with number of protons. Okay. All right. So as I said, when you're missing some spots, you aren't filled up with 8 electrons in that outer valence or outer shell, you start looking to get some electrons to fill it up, okay? So when you're doing that, you're going to try to form a chemical bond with another partner, okay? And you're going to swap electrons or you're going to share electrons. So that's what we're going to look at right now, okay? So it's that outer valence shell that will determine the bonding. All right, so we're looking here. This molecule or atom has its 2 inner electrons filled up, and it has the second shell filled up with 8. And then it's got this third shell all filled up with 8. So this is a noble gas configuration. It's unreactive, it's stable, it's all filled up. And as we're filling them up, if let's say we only had 6 in here, we would fill it up with like 1, 2, 3, 4, 5, 6, and we would leave 1 unpaired over here and 1 unpaired over here. You fill up each of these positions out here with 1 first before it gets a partner. Do you get that? So fill up 1, 2, 3, 4, 5, 6, and you'd have this position and this position out here unfilled. We'll see how that works a little bit more. Whoops. All right. And you can go across the periodic chart, like I said, and you can add in the electrons and know that the protons, you would have the same in the core. So you'd have 1 proton in the core here for hydrogen. You have 1 electron outside. Helium, it's 1s shell is all filled up with 2. It's happy. It doesn't want to react with anything. Okay. Lithium, you go in and now we're taking and we're going to add another shell outside here. So it's, you assume the inner core 2s like helium is filled up on the interior here. And now as we go into the outer shell, we're adding in 1 electron. Well, how many electrons does it take in this shell to be happy?
8.
8, okay? So when we start looking at lithium, what do you think is it's going to be easier for lithium to do? Is it going to be easier for lithium to get 7 electrons, or is it going to be easier for it to donate and give away an electron?
Donate an electron would be easier.
Donating electron will be easier. So that's the type of bonding you're going to find that lithium will do. And that's why typically you'll see lithium often written as lithium 1+, because lithium is going to try to get rid of this electron because it's just excess baggage and it makes it hard to fill up that outer shell. Same thing going on here for beryllium, okay? It's got 2 in it. So it's going to be easier for it to get rid of 2 electrons than it is to gain 6. When you start getting in here to boron, carbon, and nitrogen, things get a little bit funkier, and we'll see they end up liking to form covalent bonds and sharing electrons and partially filling up rather than giving up or just kind of stealing an electron. If we get down all the way here to fluorine, fluorine has 7 electrons and it wants to look like neon. right? So to get an extra electron, it needs an extra electron. Lithium over here is willing to give it up. And you'll see that a natural salt that likes to form or ionic bond is going to be between lithium and fluoride, where lithium will completely donate its electron to fluorine. So then lithium becomes lithium 1+ and fluorine becomes fluorine 1-. And they like to stay close to each other because now they're oppositely charged, right? So those opposite charges attract. So even though lithium completely gives up its electron to fluorine, they still like to live right next door to each other with their opposite charges, keeping that magnetic attraction of those two atoms together. And that will be called an ionic bond. And you'll see that all these elements in column 1 like to form the same kind of arrangement with all these molecules over here or atoms over here in column number 7. The other natural one that we know so much about is sodium and chloride. They work the same way, okay? All right, so you can think of these in column 1 as being the +1 ions in ionic bonds, and you can think of these over here, fluorine, chlorine, bromine, iodine, as being -1. Okay, they've all tend to gain an electron. And these will be over here what we tend to call the more electronegative atoms, and these are more the electropositive. And we're not going to be concerned about all these crazy transition metals in between, say, thank God, right? All right, we don't want to get into that chemistry right now. All right, so we'll look at all that. And then these ones right over here, this ends up being the domain and cluster of forming all the covalent bonds of our organic chemistry molecules. And we'll see what that is in a minute. Okay, so how we're going to start forming compounds. Compounds are going to simply be molecules that contain 2 or more types of atoms. And a typical compound that we form is water. We get 2 atoms of hydrogen and 1 atom of oxygen. And so we write the formula as H2O, okay? 2 hydrogens combined with 1 oxygen. And as I said, we can also have our molecules form by ionic bonds. And this is where we shared our protons and electrons. The number of protons and electrons when we start out are equal in an atom, and we make an ion when we gain or lose an electron. So in the case of lithium, or in the case of sodium, we lose an electron, right? And this becomes positively charged. So we call that now a cation, okay? Chlorine, or fluorine, in the case of when we were doing lithium fluoride, gains an electron. And this is also an ion, and we call it the anion when it gains the electron and has the negative charge. And then you'll see the cation and the anion like to stick together and form an ionic bond. Okay, so cations lose the electrons, become positively charged. Cat, the word cat has a plus in it. That's your mnemonic device for remembering which one is the positive charge. Anion, no T in it, no plus sign. It is the negative charge ion. Right, so then our ionic bonds, like I said, will form from the opposite charges that results when one atom loses electrons and the other gains electrons. Now in your textbook, it kind of, you know, some of these textbooks that end up being written by biologists, they forget some of their chemistry a little bit every now and then. And they write that the covalent bonds are stronger than the ionic bonds, but that's not quite true. All right. The bond energy that keeps your salt together, sodium chloride, is exceptionally strong. And sodium chloride has a very, very high melting point. But see, biologists are always dealing in, you know, biological matter. So everything is already solvated in water. And what happens, as you will see when we get through, is that as soon as you put sodium chloride in water, the sodium chloride dissociates or breaks apart in solution. So that's what they're trying to say, is that that doesn't happen with covalent bonds, okay? But I don't want you to get the wrong idea that sodium chloride itself is not bonded strongly and that that ionic bond isn't strong. It's a very strong bond that holds sodium chloride together, and sodium chloride could sit for eternity, not changing, not decaying, you know, on your shelf, and probably does somewhere in your shelf in your kitchen. Okay. All right. So we've already seen that we made our sodium cation, right? Because we ejected that one extra electron that we had out here in the third orbital. And then chloride got that one extra electron right here. And so it's the anion. And together they form this close relationship. They're not sharing here. They're just sitting next to each other, kind of in a magnetic attraction between their opposite charges. Right? Same thing happens, like I said, with lithium fluoride. When we look at the two valences here, we have lithium with its one extra electron in this extra outside orbital. Lithium wants to make itself look like helium, okay? It wants to get rid of this whole extra orbital or valence out here, get rid of this electron, look like helium. Fluorine over here wants to look like neon. And you see when I said that you fill up your orbitals, so you're going 1, 2, 3, 4, 5, 6, 7. You have one kind of lone one sitting up here. That's where we're going to fill it, okay? So lithium will ultimately donate the one electron over here to fluorine, and it will become lithium plus, and fluorine will be fluorine F minus, okay? And so lithium has this one extra electron, and I know these slides are repetitive, but this is so you can go back to it and look at it at home, and fluorine is going to pick up the extra electron. Before it gives up its electron, we write it, we can write it as a Lewis dot structure, and you can write what the outer orbital looks like. And this is usually easy for a lot of people. So you see, you can write lithium with its one extra electron out here. And you can see fluorine with its 1, 2, 3, 4, 5, 6, 7, and then the 8th spot being unpaired right there. And that's where lithium will donate, and it will fill in that unpaired spot, okay? And this is exactly what happens, is it transfers the electron right there, and then lithium fluoride forms because lithium is now positively charged and fluorine is negatively charged. All right, so let's move on to covalent bonds. Our covalent bonds form when atoms share electrons rather than one giving the donor. And as I said, the textbook will tell you that covalent bonds are stronger, but it's just that covalent bonds don't break apart when you put them in water the way that, or dissociate into the separate ions the way that a salt or ionic bond like sodium chloride does, all right? But a bond strength is really based on how many joules of energy it takes to separate a mole of a mole, or Avogadro's number, 6.02 times 10 to the 23rd numbers of molecules of sodium chloride into its separate sodium and chloride ions, okay? Whereas you can see two hydrogens bonded to each other covalently takes actually less energy to separate. So it doesn't have as strong a bond as sodium chloride, technically in chemical terms. But as I said, biologists care about how everything acts in the solvent of water. But I just don't want you to get confused about that. All right, so now covalent bond formation. Hydrogen, as we know, has its one extra, one electron hanging around in that S valence, okay? So hydrogen can act like lithium when it gets around a fluorine or a chlorine. But, you know, hydrogen also acts differently when it gets around another hydrogen. And so you see, when you have two hydrogens and they're exactly the same, is one going to give up one electron over to another hydrogen? And are you going to have an H plus forming a bond with an H minus? Well, it doesn't really make sense, because neither one of these hydrogen molecules with their little extra electron floating around out there is going to be more inclined to give up its electron to the other hydrogen than is that hydrogen inclined to give it up. You know, they're the same molecule. So they're kind of having about the same level of attraction to their electron. Each one of these has the same inclination to either give it up or not give it up and grab onto another one. So what they do, is you find, is they decide to share, okay? And this is how a covalent bond forms. And what happens is then this electron pair kind of swishes around and spends as much time around this hydrogen as it does moving around this hydrogen, so that electrons form in a cloud around the whole molecule and moves around both of the hydrogen cores. What you also find is that with these molecules like carbon, you see here carbon has 6 electrons around it. So it's 2s, its s orbital is filled up already with the 2, but then its next orbital has 4. So is carbon going to give up 4 electrons and become a cation? Or is it going to gain 4 electrons and become an anion? It's kind of right in the middle there. So it's not really inclined to either give up or just completely steal. So what carbon does is, and this is why when we get into the idea of organic chemistry and why so many of our compounds are based on carbon, is the carbon is the one that's in the unique position of forming a covalent relationship at each position around the whole molecule. So you'll see carbon likes to form methane with all these hydrogen atoms. They're all happy sharing their electrons together, okay, and filling it up. And it ends up being non-charged. So this is what we call a non-polar covalent bond. We're going to have equal sharing of the electrons around all the atoms in the molecule, here or here. There's no reason these electrons want to circle more around carbon than they do around hydrogen. They spend their time circling around each part of the atoms in the molecule. But now we have a new kind of critter here called our polar covalent bonds. And this is where we're going to have some unequal sharing of electrons. And this will be because one of the atoms is more electronegative and it has a disproportionate pull on the electrons. So let's go back and look at our charts over here, our periodic chart. When I was showing you over here, this is kind of like the electronegative area of the periodic chart, and this is the electropositive. These big electronegative atoms over at this corner, so if you think of an arrow or a vector going up in that direction, these atoms over here in covalent relationships are going to pull to have the electrons circle and dance around them a whole lot more. So you'll see oxygen is one of these atoms. Yes, Sarah. Sarah, you raise your hand. Trying to get her to unmute. Sarah, I can't unmute you. well, we'll move on. Okay. So all these atoms at this end of the chart are much more electronegative. So when they form covalent relationships, what you have to think of is that all of these atoms here in covalent relationships, you know, like fluorine, chlorine, oxygen over here right next to them, sulfur, they're a little bit more over at this end, and they're almost all filled up with electrons. So they kind of more strongly pull the electrons toward them. They have that positive, highly positive nuclei down in here. So they're going to strongly pull those electrons toward them. Okay. So let's get back over here. Sarah, can you ask your question? Are you unmuted yet? All right.
She could always put
Right
it in
the
chat.
Yeah, there's nothing in the chat. So Sarah, if you go over and hit on the chat, you can write in the chat column too. Just might not know the how to get that. So all right. All right. All right, so polar covalent bonds are going to have this unequal sharing of electrons. And how I like to think of it is you have your ionic that's completely an attraction between the molecules, that one completely steals away an electron from another. They're in kind of a codependent relationship, right? You know, and one's a taker and one's a giver. Our non-polar covalent bonds are the relationship that we're all looking for, where we're all happy dappy, equal sharing, everybody is all accepted. And our polar covalent bonds are, you know, you got one dominant person and one sort of non-dominant person. You're sharing, but the sharing is a little bit unequal. You know, somebody's doing more of the housework, maybe somebody else is earning more of the money or whatever, right? Okay. So this is kind of the rainbow of going across from your non-polar covalent bond. Everything is equal sharing. Your ionic bond over here, all the attraction is because somebody's completely plucked away an electron, and now you're left with opposite charges. And our polar covalent bond is, we're going to have sharing, but that bond of the sharing is a little ionic-like. You know, the electron is going to dance a little bit more around the bigger electronegative atom in the relationship. And you'll see what we mean hopefully in a minute. So now we're going to do water. So we see our hydrogen atom right here, all right, is going to hook up with our oxygen, and our oxygen has all these paired electrons over here. So highly electronegative. You can think of when you see two lone pairs of electrons out here, thinking of that as almost like a partial negative charge out there, all right? And so the hydrogen atom is going to come over here and it's going to start to form and share up here with this oxygen atom. So you see that's what happens, hydrogen sharing and sharing over here. But you still have these big electron pairs out here. And we say these electron pairs are partially negative charge. So that's where we write this, this delta negative out here, okay? So if I were to go back here and draw this in, I would say this up here would be delta negative, partially negative, delta meaning partial, not a full negative charge, but a partial negative charge. And this is going to mean this is going to draw these electrons to spin much more around the oxygen atom and not so much around the protons or the hydrogen part. So these hydrogen molecules right here, or hydrogen atoms, become really almost like what we call a proton, and they end up with partial positive charges, okay? Delta positive out here, partial positive charges, all right? I hope you all get that. And that's what they wrote in here on the charge. Okay. So now that we know what a polar molecule looks like, we can see that we're left with these areas at which we have some partial positive and partial negative charges kind of out there and exposed. And because of that, with these big electronegative atoms like an oxygen or a nitrogen, we can see that they are left with these partial negative charges. And because of that, that polar relationship, they can form little, especially when there's a lot of these molecules, weak electromagnetic attractions between partial positive and partial negative molecules, just kind of like an ionic bond. So what we see in water, when we have one water molecule and another water molecule and another water molecule and another water molecule, and if you have a whole big bottle of water molecules, what they do is they're going to line up alongside of each other. And that big partial negative charge on the oxygen is going to line up and face against the partial negative charges on the hydrogens. And the partial negative on the hydrogen or exposed proton here is going to line up against the partial negative charges on the oxygens over here. And you see each water molecule aligns like that, and the attractions kind of stay so that these water molecules lined up alongside of each other kind of are a little bit adherent to each other. They're not in a formal bond, ionic, and they're not in a formal bond, covalent, but they're in this kind of partial ionic relationship to each other. And we call that a hydrogen bond, okay? This hydrogen's facing whatever electronegative element they have. And this is what confers so many of the special properties that we see with water. So when I pour out water, let's see, what do I have here? I got a coaster. If I pour water a little bit here on a coaster, let's see if I can get this so that you guys can see it. Probably not so good. Can you guys see the water on the coaster? But if you do it yourself, what does water do when you drop it out onto something? What happens with water? Does it just flow all out everywhere, or does it kind of bead up? Like, what does water do out on your car when it rains? Does it bead up, or does it just flow all off?
It beads up.
It beads up. We know that. And this is because of the hydrogen bonds. The waters kind of are attracted to staying together, okay? You're going to have a lot of it flowing to have it flowing all over. It's going to bead up, all right? So we'll see how many of the properties of water cause it, to kind of bead up. Or when you have a capillary tube with water, and remember how you're always taught to read like in a graduated cylinder, it kind of forms like a little bit of a U shape because the water creeps up the side of a bottle. That's because of the hydrogen bonding in the water. They're all attracted to sticking together. It gives it like this stickiness property of the liquid. All right. So we can see water molecules in water, in a big tub of water, all orient and align up to each other. And you can just really stare at that and take a look at how the hydrogen bonds form. Same thing would happen with ammonia, and the nitrogen, the lone pair of electrons that you'll find on a nitrogen will also align against and have a partial charge against the partial positive charges of the hydrogens, okay? But where we really care about it is with ammonia. So let's see what we're learning. Which type of bond is formed by the sharing of electrons between atoms?
Covalent bonds.
Exactly. Right, both oxygen and neon are gases at room temperature, but oxygen combines readily with other elements, but neon does not. Why?
A.
A, neon's filled up. Its valence is completely filled with 8. Oxygen, we know, has only 6, okay? And it's because of unfilled valence positions that you form the bonds. Which element commonly only has a proton at its nucleus?
Hydrogen.
Hydrogen. And that's why you'll so often hear hydrogen referred to as a proton. Okay. The innermost electron shell in an atom holds up to how many electrons?
B.
2.
The innermost is 2. Yep. 8 is the outer ones. Okay. The outer ones beyond that. And the smallest stable unit of matter, once again, is the
Atom.
Atom.
okay. All right. Atomic weight. Let's see if you can remember the difference of atomic weight and atomic number. So which makes the atomic weight?
D.
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 B, the neutrons?
D.
G.
No, B.
D.
D,
Neutrons
yeah,
and
the protons and the neutrons, but not the electrons. Okay, not the electrons. All right. A nucleus consists of, what's in the nucleus?
D.
Protons
and
neutrons.
Protons and neutrons, same answer. Yep. And isotopes of a different element will differ in the number of what?
Neutrons. Or new. Yeah.
Yep. Okay, good. You guys get it. All right. So when electrons are transferred from one atom to another, and the two atoms unite as the result of opposite charges being attracting, it is what?
Ionic.
Ionic
bond.
bond.
An ionic bond. Yep. Okay. Not just an ion. It's neither just a cation or ion. We're talking about the bond. So that's tricky a little bit there. All right. If an isotope of oxygen has 8 protons, 10 neutrons, and 8 electrons, what's its mass number?
18.
18. Yep. So this is another way, that you can ask the mass question, 18. Okay. Because electrons don't count for mass. What's its atomic number? Atomic number is just the number of protons. So atomic number would be 8. Okay. All right. Chemical reactions. All right. So now we've kind of looked at our elements and all that. And now we're going to, oh, my internet is unstable. Shoot. I got to make sure they got the internet off up here. I'm sorry. Hang on. I'm going to make sure that nobody's running the internet upstairs. Hey, can you guys make sure the internet's not going on up here? Can you turn off, Kathy, if you got your phone going or anything? Make sure you don't have the internet or phone going. It's messing with my internet downstairs. Okay, my phone's not, but I will unconnect my phone is, well, I'm not. All right. If Jerry calls you. Yeah, if he calls my phone, Okay. All right. All right. Sorry, guys. Trying to make sure. Are we back yet? Can somebody talk to me?
Yeah.
All right.
Yeah.
All right. So now we're going to make some chemical reactions. Okay. So when we do chemical reactions, we're going to have a change in chemical energy occurring as atoms, molecules form compounds and swap partners and all that. Our bonds are storing energy. So like a single bond, when we look at covalent bonds, stores less energy than a double bond, than a triple bond, okay? When we are storing and building bonds, these tend to be endergonic reactions where we're absorbing energy and storing them in our bonds. When we have reactions where usually we're breaking bonds, we're releasing energy, and these are called exergonic. So synthesis reactions, where we're going to take 2 molecules and combine them with a bond to form a new, larger molecule, will typically be endergonic. And we call it also anabolic. Anabolic, we're building. Think anabolic steroids. Those guys build and buff themselves up, right? When we decompose a substance and we take A and B and we split it apart into its components, A and B, we broke a bond, we're releasing energy. That tends to be exergonic, and we call it catabolism. We're decomposing things, all right? So let's look at a decomposition reaction. We break AB into its component parts, okay? The reaction's going in this direction. Pretty often in our bodies, since everything is solvated in water, we use water to break apart molecules, and it's called hydrolysis to break those bonds. So here we have a substance, you know, just 1, 2, 3, 4 atoms. We use some water to break it apart, and we split off this last bond right here. So we end up attaching a hydrogen to one and an OH to the other, okay? So they look the same kind of here at both ends. That's a hydrolysis reaction. We can also have exchange reactions. We're not really, you know, completely decomposing or not really synthesizing. And in fact, we are in part doing a partial synthesis and a part decomposition, because we decompose this original partner and then we do a synthesis over here when we swap and make a new partner. Okay, so that's an exchange reaction. We've just swapped partners. So we go from AB plus CD to AD and CB, swapping partners. First got to decompose the original couple and then synthesize bonds for a new partner. We also talk about chemical reactions in terms of whether they're reversible or not. And when they're reversible, you see that we put the arrows in both directions. Now, typically, sometimes you can even see like an arrow might be smaller in this direction and larger in this direction, meaning it favors going to forming AB rather than CD. But you'll see, and in your textbook says that when the concentration of a product gets above a certain threshold, we get to a point of equilibrium where we're forming each side of the equation. They're just kind of converting back and forth. We're not really changing the relative concentrations of whether we have AB or CD. And you'll see like a common reaction that does this in our body is when we have carbonic anhydrase, which is an enzyme that will convert carbon dioxide and water into carbonic acid, okay? All right, and sometimes you write the conditions. You might write that it takes heat, or you might write that it takes an enzyme like right here above the arrows in our reactions. All right, so now let's talk a little bit about some of our important biological molecules, and let me see where we are on time. 10:21. Okay, we're supposed to go till 11, so goodness, let's try to get through this. Organic compounds always contain carbon and hydrogen, and these tend to be structurally complex molecules, whereas our inorganic compounds are things like sodium chloride. They don't tend to have carbon involved, and they're usually very simple compounds. And water is our inorganic compound that we're most concerned with. We need to realize it's inorganic. Remember that it's polar. It has that unequal distribution of charges, and it's our universal solvent. Everything in biology is pretty much solvated in water, and we'll show you what solvation means of the polar substances undergoing dissociation and being surrounded in a shell of waters. The cool thing about water is all those bonds with all those hydrogen bonds formed between the water is that it absorbs a lot of heat, and that's why it serves as a temperature buffer in our bodies and, you know, throughout the biological systems. Okay, so it's going to take a lot of heat to separate water's hydrogen bonds. That's why when you're going to make your pot of spaghetti and you stick your water on the stove and you flip up the stove and you're staring there and you're staring at the pot of water and you're like, why the hell won't this boil? I'm hungry. You know, it takes a long time to separate all those hydrogen bonds, okay? It has a tendency to want to stay liquid, okay? All right. because of all its ability to absorb and release heat, like I said, it will moderate our temperature changes. So as we put energy into our biological system, we can shift that balance between the hydrogen bond formation and the breaking of hydrogen bonds. And so if we start getting heated up in our biological system, we might shift toward breaking some of these hydrogen bonds, but it's going to take a while to do. And when we do that, we get evaporation, and that cools off our body as the water leaves the skin surface or evaporates off the surface of a plant. And of course, the opposite happens if we allow the water to keep absorbing into its bonds, right? So as molecular motion decreases and the temperatures drop, there's going to be less of a tendency to break apart those hydrogen bonds, and all the bonds become intact and they become even more stable, and they form a lattice-like structure and become solid or ice, okay? And as we know, it actually, even when it's forming those solid lattice, the molecules are actually a little bit farther apart, which is why ice is less dense. The separate water, water atoms is less dense than liquid, and ice then floats on the surface of water. It floats on lakes, it floats on ponds, it'll even float on the oceans. And this is what allows water to create life on Earth, is that the water keeps that insulating barrier on the top of the ponds during all the cold months that allows things to move along and respire and sort of be blocked off from the cold temperatures above it by the insulating function of ice. And that's why when we have the Mars rover, we're constantly going and searching for water, because we know if there is water on another planet, there's very likely the ability for there to be life. Speaking of which, have you guys seen all this crap about the UFOs in the news? And that the Navy is now releasing all these things about Navy pilots that are have been seeing UFOs, and they started releasing all these pictures that the government has kept buried for years. So I'm kind of interested in that about, is there other life amongst us? All right, so all this goes back to, somebody want to comment on that?
So they've been here for centuries, right?
I don't know.
I want to believe.
Yeah, you want to believe in it. Maybe, if you watch, what is it, the Learning Channel? The Learning Channel, they always have all those documentaries on space aliens having been amongst us for, the millennia or something. It's kind of cool, Star Wars come to life here, right? So, all right. So all this comes back to our hydrogen bond formation, which is the result of this polar molecule, with the oxygen attracting more of the electrons around it, and the hydrogen having the electrons stripped away from it so that it has these partial positive charges. And then they form the bonds with similar molecules in water, forming all those hydrogen bonds to each other. All right. Now, how does then water act as the solvent for things like sodium chloride? Well, sodium and chloride, when you put it in water, will break apart into its component ions. And so you'll have your small cation of sodium over here and your larger anion of chloride, and all the water will align its negative charges of the oxygen around each sodium, and the positive faces of all the hydrogens will align around the anion of the chloride. And this will happen over and over again in the tub of sodium and chloride dissolved in water. And that's kind of a solvation shell, okay? We call it a solvation shell. All right, so this goes through that of water, and what it is a solvation sphere or shell or sphere of hydration. There's different names for it. And you can see all the hydrogens aligning around each sodium, and all the hydrogens aligning around each chloride, and all the oxygens aligning around each sodium over here. All right. All right, so in water, In a water molecule, oxygen forms hydrogen bonds with 2 hydrogen atoms to make an individual water molecule. True or false?
False.
False. Yes, this is false. The water molecule itself, each hydrogen is in a covalent relationship to the oxygen. That's intramolecule, within the water molecule itself. Okay. Now, if I ask the question in a water molecule, between 2 water molecules, the oxygen of one molecule forms a hydrogen bond to the hydrogen of another water molecule. True or false?
True.
True. Okay, good. So you got it. All right, and then we have our acids, bases, and salts. So acids are going to be substances that when we throw them into water, they're going to dissociate into protons and some other negative ion, okay? And so we're going to call them proton donors to the solution. So hydrogen chloride, hydrochloric acid, when we stick it in water, it's going to break apart into H plus protons, because that's just what it is a proton stripped of all of electrons, and the chloride ion. And those H pluses are what makes it acid. All right. Bases are substances that dissociate into hydroxide ions, okay, which are anions, and 1 or more positive cations. So if we say our typical base of sodium hydroxide, okay, right here, breaks apart into sodium cations and the hydroxide ion. And the whole point of bases is they can accept that proton, right? They react together and they will form water, H plus this hydroxide ion. All right, so HCl thrown into water, breaks apart into H plus and Cl. You see the H plus, you know it's an acid. Sodium hydroxide breaks apart in the water into sodium and this hydroxide ion, and you know it's a base. It's a little bit more complicated, but this is good enough for our purposes. And together, all these acids and bases, you can also have salts. Salts are going to be substances that when you put them into the water, they dissociate into substances that are neither a proton nor the hydroxide ion. So we put sodium and chloride in the water and we get our sodium cation and our chloride anion. And neither one of these is a proton. Neither one of these is a hydroxide ion. So it's a salt. It doesn't affect our pH. Okay. All right. So there's our salt in the water. What is our pH? pH, the P just means the negative log base 10. Okay, this is math, and so it always makes a certain number of you want to have your head spin. But what you need to think of is that as the H plus concentration increases, you're making it more and more acidic. We just put things on a logarithmic scale to make it so that we can see things nicely in a graph, okay? So as we increase the amount of H plus, we're increasing our acidity, we're going to end up dropping our pH. As we increase the amount of OH minus, we're going to be increasing the alkalinity, and we're going to be increasing the pH. It's on a log 10 scale, and I'll explain what this means as we get to the next slide. But know that most of the organisms, all these microbes that like us, are going to want to grow best between pHs of 6.5 and 8.5. Our pH is somewhere around like 7.2, 7.3. And as I said, there's always going to be a tropism, okay? The tendency of the microbe to want to grow somewhere specifically. And they're going to like a specific temperature, they're going to like a specific pH, and so they're going to like that specific pH in your body to grow at, and the conditions. So when we talk about the pH scale, that logarithmic log of base 10, as you go on the pH scale, 1 notch each time, it's changing kind of like the Richter scale. by 100. So the difference between a pH of 1 and a pH of 2 is logarithmic scale. I'm sorry, it's by 10, okay? It's by 10. This will be from 1 to 3 will be 100. From 1 to 4 will be 1,000 times. So 4 is 1,000 times, or 10 to the 3 difference from 10 to the 1, okay? Down here, difference in wine or tomato juice from stomach acid. The lower you are on the pH scale, the more acid you are. Right at 7, neutral is where the amount of H plus equals the amount of OH minus. When we get into there being more OH minus than there is H plus, then we're up here in what's called the basic scale. All right. So, and you can see milk, pure water, human blood, we're all right here around neutral. Okay. Just within a couple of notches around 7. All right. So organic compounds are going to be compounds that contain hydrogen, oxygen, and nitrogen in addition to carbon. Organic chemistry is really the chemistry of carbon, if you haven't ever taken organic. It's not the same meaning, of course, as organic that we use in parlance for talking about organic food or buying organic, things of your organic food, your organic shampoo or whatever like that. That's usually meaning natural and without preservatives and such like that. All right, so we're referring to organic chemistry as the chemistry surrounding a carbon of skeleton. So attached to our carbons will always be in these covalent relationships. We'll have functional groups. So functional groups right here, this will be whenever you see OHs, these are hydroxyls. We've already learned about like the hydroxyl ion for our pH scale. And these are called alcohols when it's just an OH hanging off here, okay? Methanol, ethanol, okay, propanol, propanol. We have different functional groups. So you have your alcohol, you have an aldehyde when you have a C double bonded to an O and an H at the end of a molecule. When you have your C double bonded to an O in the middle of a molecule, it's called a ketone. They're very related, alcohols and ketones. You have a methyl group when your carbon is completely hydrated, has hydrogens all around it. When your carbon is connected to an NH2 group, that's called an amino, okay, which you see in amino acids. When you have your carbon with a double bonded O, and then it's got a single bonded other O connecting to other carbon groups, it's an ester. Going on with an ether, we have sulfhydryl. I don't have to go through all these. The other one that you want to be concerned about is carboxyl. The COOH, you'll see it written as a lot. It's one double bonded O, and then also single bonded to an alcohol or hydroxyl group. All right. So we learn our functional groups, and we have to identify them when we look at our organic compounds. So we form our amino acids. So our central carbon right here has an amino group off of it, and it has a carboxylic acid, that COOH group, amino acid. All right, so we're going to be combining some small organic molecules to make larger macromolecules. And most of our macromolecules will be polymers of many repeating monomers. Okay, so we'll repeat like simple sugars over and over into polysaccharides from monosaccharides. And we'll see that our bodies and chemistry and the microbes will do these over and over again. Common themes being we synthesize by removing water, which is called a dehydration synthesis, to join substances together. Loss of water, condense them, like your condensed orange juice, loss of water, dehydration synthesis. Okay, So we're going to take A and B, and we want to join them together to make AB. And we do it usually by removing water somewhere off the end of the molecule. So if this is A, and it's got this carboxylic acid group, and we're going to combine it with this amino group over here, we're going to remove the OH and the H. So we're removing water, and we're going to form a new bond between this C here and this N here, right? And so we now have the amino acid, and we've removed water as a condensation or a dehydration synthesis. And you'll see that we do that over and over again, dehydration synthesis, when we're forming bonds to join our monomers of our sugars together. So the simplest of our carbohydrates are going to be simple sugars. And they consist of carbons, hydrogens, and oxygens with a typical formula of CH2O, hydrated, okay? That's where you got hydrated carbons, right? Carbohydrate to the end, multiple times over. And you can see many of the carbohydrates are isomers of each other, which just means the carbons are rearranged differently. That you can see these all have the same chemical formula, but they're just rearranged differently. So the simplest are our monosaccharides, glucose, fructose, galactose, okay? And they're isomers, they're rearranged differently. Disaccharides we form by combining one sugar to the next sugar. We do a condensation or a dehydration synthesis going in this direction, and we form the new bond here between them, an ester linkage, okay? Removing water. Of course, if we wanted to break it apart, we're eating our candy bar, we want to break down our sugars and our body needs them. We're going to hydrolyze this bigger sucrose into its component parts and put the waters back on the ends of the molecules when we break apart the bond. So dehydration synthesis, building up, anabolism in one direction, catabolism, breaking it apart, hydrolysis in the opposite direction. And so we can build them up from beyond disaccharides into polysaccharides, big chains of the same carbohydrates over and over, multiple glucose molecules linked together, forming starch. And starch will have it going off, even forming chains in different directions. We also have lipids, okay? Lipids, primary component of our cell membranes, right? Our phospholipid bilayer. So you're going to have phosphates attached to lipids, all right? If you had a question somewhere along the way about putting radioactive phosphate into your body, where do you think it's going to show up? In every phospholipid bilayer membrane, right? Okay. All right. So consisting of carbon, hydrogen, and oxygen, but it doesn't have that same formula of CH2O multiple times over. It's not carbohydrate. Okay. So you're going to find that it's going to have a lot lower oxygen content. Okay. A lot lower oxygen content. Probably should have put that on that slide. And so our lipids are going to have simple formulas. We're going to have fats or triglycerides, and we're going to be combining fatty acids onto glycerol. So you have your glycerol molecule up here, and you can see that when we want to attach a big long fatty acid to it, we're going to do another dehydration synthesis and remove this water right here and attach it to it. All right. So fatty acids are just long chains of carbons that are hydrated with hydrogen, right? Hydrocarbon chains, all right? So when we make our complete triglycerides, we have 3 fatty acids attached to the 3 carbons of glycerol. And we removed water here each time we did it, attaching that. And so here we have 3 different fatty acids on to this glycerol. We have one that's just a long chain of carbons that are hydrated. Here we have another long chain of carbons that are hydrated. But here we have a carbon that is double bonded to a carbon. So it's incompletely hydrated. It's got this double bond that throws a kink in it. And we call that an unsaturated fat when we throw in a double bond. It's called a cis fat when both of the hydrogens are on the same side of the double bond. But let's say if the hydrogen over here was on this side of the double bond, it would be called a trans fat. Okay. All right. And we can make our lipids more complicated, as I said, by adding other molecules to them like phospholipids, nitrogenous lipids, sulfated lipids. Okay, phospholipids are the ones we're always concerned with because they're the main component of our cell membranes, both our cell membranes and those of the microbes. Okay, and so here is our phospholipid. So we have some of our fatty acids hanging off here, and instead of on the triglyceride having all our fatty acids, having a third fatty acid down here, we have a phosphate group thrown on it, okay? And this phosphate group has like this negative charge, so it's polar, okay? We've now made a polar molecule. All this down here is non-polar. This is just a bunch of carbons and hydrogens. So we find in our membranes, and how we typically draw them is we put our polar heads of the phosphate group and the non-polar tails of all these long fatty acids. And they line up together like this, forming an interior of all the non-polar fatty acid tails. And on the outside, facing the external environment, we have our polar heads, but we also have polar heads facing the internal environment of the cytoplasm. And then it's always the complicated thing where we have to get into talking about whether we're going to do active transport, passive transport of what molecules can move in and out of the cell through our phospholipid bilayers. Okay. All right. When we make our phospholipid bilayers, we're also going to see that interspersed in between, we're going to have some sterol molecules, usually cholesterol. Cholesterol gets a bad name, but it is necessary for life. We need it in all of our cell membranes. All right, so cholesterol is also a type of fat. It's called a steroid. And a steroid basically has a nucleus that looks like this, where you have these 3 6-carbon rings and this 5-carbon ring like this, and has this kind of open structure that looks like it should be 2 6-carbon rings. And you can see here is cholesterol right here. And all of our steroid hormones are made from cholesterol and have the basic same structure. That's getting into biochemistry, which this class is not, so don't worry about it. Just kind of recognize that. Our next macromolecule is proteins. And again, our proteins are going to be made of carbon, hydrogen, and oxygen. And now we bring in nitrogen. So if we had a question about, I wanted to put in a bunch of radioactive nitrogen, what's going to take it up? As we talked about earlier, we're going to see it in things that are nitrogenous compounds, like the urea in urine, or like the nitrogen in proteins. We'll also see there's nitrogen in our nucleic acids, so they might take it up too. All right. So proteins are essential in all of our structures and function of our cells. And enzymes are some of our primary proteins, okay? We're making enzymes all the time to help speed up all these chemical reactions. How do you know something's an enzyme? Just by looking at it and reading the name.
It has the ASE ending.
ASE ending, right? Yeah. Speedy upase, right? Catalase. Catalase is going to be an enzyme you'll see with your microbes a whole bunch, okay? Carbonic anhydrase, okay? These are all enzymes. Urease, okay? Splits urea. Okay, so other proteins that are going to be important will be transporter proteins that will help move chemicals across those membranes, okay? They'll have little channels that open up and allow things in. We'll see a lot of our microbes will have proteinaceous appendages that hang off of them called flagella that will kind of propel them, spin around, and help them in their movement. And we'll also see that some of the our proteins in our bacteria are going to be toxins. Okay, some of our bacteria will secrete some really bad toxins that can be deadly to us. And some of them will even have what's called an endotoxin that's part of their cell outer membrane. All proteins will consist of subunits called the amino acids. The amino acids will have the acid component, a carboxyl group, and the amino group that we showed before, and another side chain of hydrocarbons. So our amino group, an amine, NH2, and our carboxyl group, the COOH, and this R group is going to be some other chain of carbons, okay? Like here on tyrosine as a cyclic carbon group. Here are the main 20 amino acids found in proteins. You don't need to memorize these, but you can kind of look at them and see if you can identify the different amino and acid part, acid, amino. This acid part would combine with this amino part. They would lose a hydrogen here and the OH here and form a amino acid, okay? Protein bond. Peptide bond is what it's called, okay? All of our amino acids are stereoisomers. I don't think you really need to know that for here. It's in there from other slides. Okay, so this is again how your peptide bond forms is of course by dehydration synthesis, removing water, and then we form what's called the peptide bond between the two. And of course we could hydrolyze it and put the arrow going back in the other direction that we want to hydrolyze and break down our glycyl alanine back into the glycine and alanine component amino acids. When we talk about proteins, we always talk about 4 levels of structure. The primary structure, which is the chain of amino acids, whether it's glycine, leucine, tyrosine, alanine, whatever in the chain, the literal chain of them. And then we talk about how the proteins kind of fold back on each other. They might form different domains called pleated sheets, or they might twist around in an alpha helix. We'll also talk about a tertiary structure is beyond these simple folds, that they all fold back in big domains over each other, and they have particular patterns, and we see that's really important for things like our enzymes fitting to the substrates. And then we might have an even quaternary level where we have big molecules like hemoglobin, where we'll have one protein chain all folded up in its tertiary structure associated with another tertiary structure. We have 4 of them together associated, kind of all wound together, okay? And that's its quaternary structure. All these proteins can undergo denaturation, and that will be really important to understand. When you fry an egg, you denature a protein. You've changed the protein and it can't ever go back, okay? It can't ever unfry an egg. That's denaturation. So when proteins encounter a hostile environment, like an egg encountering my frying pan, it loses its shape and it changes its function, okay? And we'll also see that a lot of our conjugated proteins, will describe them as consisting of amino acids with other things hooked onto them. Glycoproteins, we've added a sugar onto our protein. Nucleoproteins, we've added a nucleic acid onto it, or a lipoprotein, we've added some lipid onto our proteins. And our last macromolecule is the nucleic acid. It starts out as nucleotides, which are 5 carbon sugars, a pentose, plus a phosphate group and a nitrogenous base. The nucleosides are going to be the pentose, or the sugar, and the nitrogenous base, okay, without the phosphate group. The nucleotides that we're concerned about, or the nucleic acids, are going to be DNA, okay? DNA, you know, is what makes up all of our genes, contains the recipe for making you who you are. And we have our adenine that bonds with thymine and cytosine that binds with guanine. You've learned this before, and they wind around in a double helix held together by hydrogen bonds, okay? We break apart the hydrogen bonds to unzip our DNA so that we can read it. And we'll find that the bacteria kind of use the same genetic pattern that we do. They use their genes are encoded in DNA. but we'll find that some viruses actually encode their genetic material in RNA. So they're kind of the opposite. Some are in DNA, but all right. Somebody just asked a question up here. Oh, sorry, I had to drop. All right. All right. So this slide can remind you of what holds DNA together. Is it almost time? We're pretty much at time. Let's just try to finish these slides and we'll pick up with our cell stuff next time. Oh, and before everybody goes, let you know that next Thursday, I want to start meeting at 9 o'clock. So we kind of catch up because we lost a little time. We were going a little bit slow the first week because of everybody, doing adds and drops. I didn't want to get too much into this complicated stuff when we didn't have everybody here. Okay, so RNA is the other nucleic acid we want to be concerned with. RNA is DNA stays in our nucleus in eukaryotes. It doesn't leave our nucleus. RNA is what will leave our nucleus and take our DNA message out to the rest of the cytosol to make our proteins. Okay, it carries the recipe for the proteins. RNA is single-stranded, and instead of adenine bonding with thymine, it bonds with uracil. All right, and remember, some of our viruses are going to encode their genetic code in RNA instead of DNA, but our bacteria and us use DNA. All right, and this is uracil that looks different. And this is just, you don't need to memorize uracil, but you can see the whole component of the nucleoside right here of the sugar and the phosphate group. I'm sorry, the sugar and the nitrogenous group, and then the phosphate group added onto it makes it the nucleotide. And of course, we also have ATP, okay? And ATP is going to have that nitrogenous base attached to the sugar, and instead of being attached to a single phosphate group, ATP has 3 phosphates. These are high energy bonds, and whenever we pluck off one of them and go from ATP down to ADP, we release a boatload of energy, and that's what our body harnesses to drive so many of our reactions, is taking off the bond right here in this high energy bond, splitting it there. All right, so ATP stores energy and then it releases it when that bond is hydrolyzed, okay? All right, and here you can see ATP with its 3 phosphates, water coming in, hydrolyzing off one, shooting off this one inorganic phosphate and releasing all the energy that was contained in that high energy bond right there that will drive other reactions. If you're having trouble, you can look at this Cassiopeia project for understanding chemical bonds. Of course, you can always ask me and I'm happy to meet with you and go over any of this. A lot of this, we review it pretty heavily, just enough so that you can hopefully have a review in your mind and understand some of the chemistry as we get into it here with our class. And if you're having trouble with chemistry, the Khan Academy videos for chemistry are really, really good. He has the ability to go through and he can still draw on a board. I wish I had a big whiteboard back here behind me to draw, and I should probably go out and buy one maybe. Then I need to buy a camera